Welcome to the Explainer. Today we're jumping right in to decode one of the most revolutionary scientific tools of our time, CRISPR. We're going to look at what it is, where this incredible technology came from, and believe me, how it's already changing our world.
Okay, so let's get started. And we're not going to begin in a lab or with a bunch of theory. Nope, we're starting with a life-changing medical breakthrough that is happening right now. An incredible asset to have? I mean, just let that sink in. This is how a top doctor described the world's very first medical treatment using CRISPR. It's a quote that really tells you just how big of a deal this is. That's right. In late 2023, something amazing happened. Regulators in both the UK and the US gave the green light to a therapy called Casgevy. What does it do? It offers a functional cure for sickle cell disease, a really painful genetic disorder that messes with red blood cells. This is a historic moment. It's the first time ever that a CRISPR-based therapy is actually available to patients. So, this brings up the big question, right? How on earth does this revolutionary technology actually work? How can we literally edit the code that makes us who we are? Well, at its heart, you can think of CRISPR-Cas9 as a kind of biological search and replace function for our genes. It's got two main parts: a protein called Cas9, that's the scissors part that does the cutting, and something called a guide RNA, which is what tells the scissors exactly where to go and make that cut. The whole process is actually, pretty elegant. First, that guide RNA acts like a super precise GPS, leading the Cas9 enzyme to the exact spot in our DNA we want to change. Then, Cas9 does its thing. It makes a clean snip right through the DNA. This kicks the cell's own natural repair crew into action. And that's where the magic really happens. As the cell fixes that break, scientists can either knock out a faulty gene or even slip in a new, healthy one. You might be thinking this technology was dreamed up in some sterile, high-tech lab. But the truth is, its origins are way more ancient, and honestly, a lot more surprising. This wasn't invented, it was discovered. To find the source, we have to travel into the microscopic world. And the story starts with a battle that's been raging for billions of years between bacteria and the viruses that are constantly trying to infect them. Yeah, the name itself, CRISPR, is an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. I know, it's a mouthful, but it perfectly describes this really weird repeating pattern of DNA that scientists first noticed inside bacteria. And here's the key. Bacteria are actually using these CRISPR sequences as an immune system. So when a virus attacks, the bacterium basically grabs a little snippet of the virus's DNA and stores it in its own genome. Think of it like a genetic most wanted list. If that same kind of virus ever shows up again, the bacterium uses that stored memory to recognize the invader and sends out its Cas enzyme to chop up the viral DNA. Threat neutralized. So you've got this ancient bacterial defense system. How in the world did scientists figure out how to turn that into a revolutionary gene editing tool that we can use on pretty much anything, from plants to human cells? Well, the journey from just a curious observation to a world-changing technology happened incredibly fast. It all started back in 1987, when a scientist named Yoshizumi Ishino first noticed these weird sequences. But the absolute game changer came in 2012. That's when Jennifer Doudna and Emmanuel Charpentier showed that you could program the system to cut any piece of DNA you wanted. Just 1 year later, researchers Feng Zhang and George Church proved it works in human cells, and the floodgates opened. For that incredible work, Doudna and Charpentier rightly won the Nobel Prize in 2020. All right, so now we know how it works and where it came from. Let's get to the really breathtaking part. What is CRISPR already doing? This is way beyond theory now. These are real-world applications that are changing everything. 69. That number is the number of gene edits made to a pig's genome. Why? To make its kidneys suitable for transplant into a living human being for the very first time, which just happened in March of 2024. They edited out harmful pig genes and added human ones to stop rejection. This is a massive leap forward in trying to solve the organ shortage crisis. And what about cancer? In some groundbreaking trials, scientists are using CRISPR to create hyper-personalized treatments. They're actually taking a patient's own immune cells, their T cells, and editing them to become better hunters, specifically trained to recognize and destroy that person's unique cancer tumors. It's like turning your own body into a super soldier. The applications can get pretty unexpected, too. Get this. Researchers used CRISPR to figure out how the world's deadliest mushroom, the death cap, actually kills people. And by doing that, they stumbled upon a potential chemical antidote that's already approved for other uses. And the list, it just keeps on going. CRISPR is being used to make crops that can handle climate change better. It's being tested to fix the genes that cause inherited blindness. There are even trials for a one-time treatment to permanently lower high cholesterol. The possibilities, honestly, they feel almost endless. Of course, having this kind of incredible, almost godlike power to rewrite the code of life, well, it naturally leads us to some very big and very important questions about the future. We have to weigh these amazing cures against some really profound ethical questions. A really important distinction to make is between somatic editing, which only affects the person being treated, like with the sickle cell cure, and germline editing. Germline edits are changes made to sperm, eggs, or embryos, and those changes will be passed down to all future generations. That's where things get really complicated, raising huge concerns about unintended off-target effects and the long-term consequences of changing the human gene pool forever. So as this technology keeps moving out of the lab and into our lives, the next chapter really depends on the choices we all make as a society. CRISPR has given us the pen, but the story of how we use it, well, that's still being written.